Now that we have our tubes labeled and our plates labeled, we're going to start prepping our dilution series by adding 900 microliters of water into the 10x, 100x, and 1000x tubes. You're not going to be adding any water into the 1x tube because the 1x tube is not concentrated. It's just it's not diluted. It's the pure homogenate of whatever it is that you're using for this dilution series. Then we're going to take 1000 microliters of our pure concentration of homogenate, and we're going to put that into our Eppendorf tube marked 1x. This was already vortexed before, but if it's not vortex, you should have vortex the solution. Then you're going to take 100 microliters from that pure concentration, and you're going to add it to the 10x tube. We're going to cap that tube off. Now from this 10x solution, we're going to take 100 microliters and put it into the 100x tube, but we can't do that until we have vortexed that 10x tube, because otherwise you're not going to have the mixture completely homogeneous and you're not going to have a truly 10x concentration that's going into the 100x tube. So take that 10x tube that you just added the 100 microliters in to the 900 microliters of water and vortex it till it's thoroughly mixed. And after it's thoroughly mixed, you're gonna take another 100 microliters, but from that 10x tube, and you're going to pass it to the 100x tube, right? And you're going to continue to do this. Now you're going to vortex the 100x tube. You're going to continue throughout until you're done with your dilution series, and then you'll be ready to spread plate. We're going to speed up the rest of the dilution series preparation. And once the dilution series is set, you can then start pipetting 15 microliters of the dilution series onto each plate. Now make sure that your 10x dilution series is going into your 10x plate, and so on and so forth, and that you're changing pipette tips and keeping a aseptic technique throughout. You're going to just put a little puddle in the middle for each one, and then we're going to be adding some glass beads and using the glass beads to spread the inoculum around the 15 microliters of fluid to get a uniform coating on the plate. And this is going to help pick isolated colonies from the inoculum of your choice. Now make sure to be using clean beads when you're doing this. They need to have been sterilized and autoclaved. You're going to put roughly about 5. They can be a little bouncy on the agar, so kind of just shake it in gently. Some of them might end up flying out. Just make sure that if they do, you're going to pick it up and put it into the waste jar for the glass beads. And we do recycle these beads, so just have an extra container to collect those waste beads. Then you can stack them up, and then what's really cool about using the glass bead spread plate technique is that means that you can inoculate several different plates at the same time. Here I'm shaking the plates back and forth, I mean front and back, making sure that the beads are staying on the agar, and I'm doing that 10 times, and then I'm rotating it clockwise a quarter of the way, and then I'm doing it again. And then once you're done with that, and you've gone completely around, you can toss your beads. There's another method that you can use. You can also use a figure-8 motion to get the beads to split to completely coat the plate. Both I found that both methods work pretty well. So now these plates are ready to set in the incubator for at least 24 hours, and make sure to flip them upside down so they don't condensate.